Welcome to UGC E-Padhshala Postgraduate Learning in Food Science. I am Dr. Maria Margaret Joseph, Associate Professor, Department of Home Science, Women's Christian College. In this module, we are going to study all about the objective evaluation of food quality. To understand the specific use of various instruments in the evaluation of individual parameters used in the objective evaluation of food products, this will be one of the main objectives of our study today. Another major objective would be to study the methods which are going to be employed for evaluating food quality that depend on some other measure other than the human senses that are often called objective methods of evaluation. So what are the advantages in doing these objective methods of evaluation? One, confidence can be gained as they are reproducible. Secondly, the results could be accurate. Human sensitivity is not involved. Minute differences can be noticed by doing objective tests. Thirdly, they are less subjected to errors when compared to sensory methods of evaluation. Next, these methods provide a permanent record so that comparisons can be made over a period of time. Further, they are not affected by factors other than the one being measured. Furthermore, emotional burdens and individual ability can be overcome. But the objective methods also have some disadvantages. One, it is time consuming. Secondly, it is expensive. Third, you need technical knowledge to do these methods. Fourth, instruments may not be available all the time. Fifth, some aspects of food cannot be evaluated by objective methods. Example, flavor. Usually, both sensory and objective methods are done. Objective evaluation supplement or reinforce the data obtained subjectively through sensory evaluation. There are some basic guidelines which have to be observed for the objective tests. Firstly, you have to conduct all the objective tests appropriate to the experiment for which equipment is available. For example, checking the pH or the hydrogen ion concentration of mixtures prior to and after heating. This information often is valuable in explaining results. The flow properties of batters or certain other mixtures prior to heat treatment can be measured, and that information used in interpreting and explaining results. Secondly, you must obtain necessary testing devices. In the preliminary testing phase of an experiment, you have to analyze all the steps in the preparation of the product and study the final product to determine whether there are specific characteristics that might be tested objectively, if additional testing equipment could be procured or developed, or it could be developed for the experiment. Thirdly, one should be meticulous about maintenance of objective equipment. Before using any equipment, the good researcher will check to be certain the machine is operating correctly in all aspects. Fourthly, carefully define the samples to be used for objective testing. A template of the item being tested often is an essential tool in obtaining comparable samples for objective tests. The dimensions of pastry or cookie samples being tested for tenderness on the shortometer must be identical. To obtain the sample, the thickness of the mixture prior to baking must be controlled precisely. Next, you have to establish the operating conditions for objective testing. For example, the temperature of a starch paste being utilized for a line spread test must be specific and controlled so that the effect of temperature on viscosity of starch paste is not an uncontrolled variable in the measurement. Samples to be controlled in size, storage, and temperature. Each experiment needs to be designated to eliminate uncontrolled variable in objective testing. The tests used for objective evaluation include chemical methods. These chemicals are estimated in foods for food spoilage like peroxides and fats, adulterants in food, for example, presence of starch in milk, maternal yellow in turmeric powder, and loss of nutrients during cooking can be estimated. Furthermore, physicochemical methods, which include measurement of hydrogen ion concentration, can be found by the use of pH meter. It utilizes a glass indicating electrode and a reference electrode to complete the electrical circuit. Next, digital salt meter. This refractometer has electrodes built in the measurement unit, and it indicates the salinity percentage in digits in 3 seconds with 1 ml sample dripped on it. Digital salt meter. Also, the sugar concentration can be found using a refractometer. It is used to determine the concentration of the sugar solution. Light is refracted as it passes through the sugar solution, with the specific values being calibrated in degrees, Brix, an indication of the percent of sucrose in the solution. The Brix or boiling hydrometer gives directly the percentage of sugar by weight in the syrup. It is always necessary to make a temperature correction since the hydrometers are usually calibrated at 20 degrees centigrade. Each instrument should be used by canners usually covers a range of only 10 Brix or 50 to 60 Brix respectively and are graduated to 1/10 divisions. Brix is defined as a percent sucrose measured by the Brix hydrometer. Products used at different Brix measurement range in percentage. Main samples to be measured in Brix range from 0 to 32 units. Fruit juice, soft drinks, lactic drinks. Depending on each food, we are going to analyze whether it's a ketchup or a sauce or honey or condensed milk. The unit of Brix would vary. But this is the basic unit to measure the sweetness in all these products. Since continued use of hydrometers in hot syrup affects their accuracy, they should be checked frequently by more accurate instruments. Next, we have the polariscope, which is used for quantitative analysis of sugar. We have the butyrometer. It is an instrument consisting of a calibrated glass tube for measuring the butter content of milk. The milk is mixed with a certain volume of ether which dissolves the butter. Then an equal volume of alcohol is added. The butter floats on the surface in the form of an oily layer and its thickness measured by the graduation of the tube clearly shows the proportion of butter. Next, we come to microscopic examination. Some properties of foods depend on their structure and valuable information can be obtained by microscopic examination. Some examples are given below. The type of organisms present in fermented products like idli batter, examination of starch cells under the microscope for identification, spoilage of the food can be found out by observing the organisms under the microscope. Size of crystals in sugar is related to smoothness of the product. The number and size of the air cells in batters and foams can also be detected by microscopic examination. There are other physical methods of finding out the objective tests indicating the quality of a product. They include the weight of a food in case of an apple or egg. The volume, liquid volumes can be measured by using measuring cups. Solid food volume can be found by displacement methods. In this method, the volume can be calculated by subtracting the volume of seeds held by a container with a baked product from that of the volume of seeds without the baked product. Usually, mustard seeds are used. Specific volume. The determination of specific volume of any product should be done with care. An average of replicates are to be taken since experimental errors could occur. Measurement of bulk volume in a porous and spongy product like idli is difficult. The volume may be measured by displacement with solvents like kerosene. The idli is given a momentary dip in molten wax to seal off the pores. Increase in the volume is taken as a measure of the bulk volume. Next, index of volume. It can be found out by measuring the area of a slice of food with a planimeter, for example, a slice of bread. It is important to use a slice that is representative of the entire product, such as a slice from the center of the loaf of bread. Index to volume is a measurement and of the food, and this is sharply pointed, and it can be made by using an outline of the slice of bread with the ink on a graph paper. And it can also, the grains of the bread can be outlined by stamping it in an ink pad and then placing it on a paper and further study the structure of the grains. Specific gravity is a measure of the relative density of a substance in relation to that of water. The measurement is obtained by weighing a given volume of the sample and then dividing that by the weight by the same volume of water. This technique is used for comparing the lightness of products physically unsuited to the volume measurements. Example, egg with foams, egg white foams. Potatoes with low specific gravity have cooking characteristics different from those of potatoes with comparatively higher specific gravity. Next, we come to the factor on moisture. Press fluids. Initial weight of the sample is noted and then appropriate pressure is given and the sample is weighed again. The difference between the two represents the amount of juice contained in the original sample. Example, juiciness of meats, poultry and fish. Drying. The weight of the original sample is determined and then the food is dried until the weight remains constant. This will give you the percentage of moisture that is lost. Next, we can see some other methods for objective evaluation of food. It is wettability. Baked products can be tested for moisture level by conducting a test for wettability. For this test, the sample is weighed before being placed for 5 seconds in a dish of water. Immediately at the end of the lapse time, the sample is removed from the water and weighed again to determine the weight gain. High moisture retention is synonymous with good wettability, a sign that a cake probably will be considered to be appropriately moist when judged subjectively. Cell structure. The cell structure of baked products is important characteristic to measure the uniformity, size and thickness of cell walls. Photocopies of cross-sectional slices give this valuable information. This technique gives third dimensional view to the cells on the cut surface of the sample and give the actual size clearly. The size of the grain. It can be found out using photography or ink prints with stamp pad or sand retention. Example, idli. Cut the idli into 2 pieces, take 1 piece, press it on the stamp pad and take an impression on the paper. The ink prints may be less clear but satisfactory for some purposes. Photography, this may be color or black and white, They may not represent the sample size, so a marked ruler should be kept adjacent. Measurement of color. Color is the first quality attribute a consumer perceives in food. Change of color is generally accompanied by flavor changes. There are color dictionaries of Merz and Paul, is the most commonly used, consisting of 56 charts. 7 main groups of hues are presented in order of their spectra. For each group, there are 8 plates. In place of color dictionary, color reproduced on secondary standards such as painted test panels or rings, discs or plastic models can also be used. A mask of neutral gray having 2 openings is used. The size of each opening should be equal to the size of the individual color of the path until it matches the color that is noted. Disc colorimeter. Here the discs have radial slits so that a number of them may be slipped together with varying proportions of each showing. The discs are spun on a spindle at about 2700 rpm so that the colors merge without flickering. The test sample is placed adjacent to the spinning disc under controlled illumination and both are seen simultaneously. Colored chips. It's a simple method to match the color of the food with a color chip or a color glass chart or color tiles. This method is not very satisfactory as it is difficult to match the food with one small block of color, and the data are difficult to tabulate and analyze also. Spectrophotometer is a visual matching of colors, is subject to shortcomings of human observers. This can be overcome since humanly you can have some error. You can use a spectrophotometer to overcome this human error, and the light will be differently absorbed depending on the color of the liquid and the intensity of the color. 2 liquids of exactly the same color and intensity will transmit equal fractions of light directed through them. If one of the liquid is a juice and the other is a juice diluted with water, the later sample will transmit a greater fraction of incoming light, and this will cause a proportionately greater deflection of the needle. Such an instrument can also be used to measure clarity, cloudiness of a liquid depending on the amount of light the liquid is allows to pass through the liquid. Now there are different instruments which are used to measure the texture of a product and to measure liquids, semi-solids. Rheology is a term. It is defined as a science of deformation and flow of matter. It has 3 aspects: elasticity, viscous flow and plastic flow. The science of rheology deals with the measurement of various mechanical properties of food, and it's important for 2 reasons. It determines the flow of properties of liquid foods to ascertain the mechanical behavior of solid foods when consumed and during processing. Instruments used to detect the liquids for liquids and semi-solids include the resistance or internal friction to the flow of liquids, normally known as viscosity. Viscosity or consistency is an important factor influencing the quality of a large number of food products like cream style corn, salad creams, jams, mayonnaise, fruit pulp, where the acceptability largely depends on their having proper consistency or viscosity. You can't have a very watery jam or very thick fruit pulp. Measurement of this factor of the raw material or the product at various stages of manufacture serves as an aid in checking or predicting the final product consistency. Further, such quality control measures also serve as indicators in calculating the amount of an ingredient, for example, amount of thickening agent or starch that is used, that should be added to a particular food product. Duration and amount of heat applied in a process may also be suitably regulated to some extent by viscosity measurement, as heat penetration and consistency are closely interrelated. Another parameter we can see is the percent sag. The depth of a sample, such as a jelly, is measured in its container by using a probe. The product then is unmolded onto a flat plate. The greater the percent sag, the more tender is the gel. The depth of a container also influences this property. Now, Baker's gel meter. This is an instrument also used to see the relative viscosity of pectin extracts or solutions in the ratio of the time of flow of the pectin solution through a given orifice compared to the time of flow of an equal quantity of water at the same temperature. There are other instruments to measure jelly strength. They are divided into two broad groups, and the method under the first group measure the breaking strength of jellies when they rupture after exceeding the elastic limits, while those of the second group measure the jelly strength by taking into account the deformation of jellies within the elastic limits. There are some other instruments which also can be employed to see the objective quality of food products. One such instrument is a Stormer viscometer. It is used to measure the viscosity or consistency of certain food products and to give an index of the resistance of the sample to flow. The number of seconds required for the rotor to make 100 revolutions has been used to measure the consistency of some food samples. The Brookfield synchrolactic viscometer is based on measurement or resistance to rotation of a spindle immersed in the test material. This can be successfully used in measuring the consistency of custards, pie fillings, tomato products, cream style corn, mayonnaise, salad dressings, etc. The penetrometer is a commonly used product to measure tenderness of some foods. This device consists of a plunger equipped with a needle or a cone that is allowed to penetrate the sample by gravitational force for a selected period of time. The larger the reading, the longer the distance, the more tender is the product. Gels and many baked products are particularly well suited to tenderness measurement using the penetrometer. There is also another instrument called the bar bender faranograph. This is used to measure the plasticity of wheat dough for preparing bread products. It is designed to study the physical properties of the dough by recording the force required to turn the mixer plates through the dough. The force required increases as the solution develops during mixing and later decreases as the solution is slowly broken down by over mixing. So in this module, let's recap what we have dealt with. We have seen the importance of objective evaluation of food and also the different methods which can be employed for objectively evaluating a food and with their merits and demerits. So with this knowledge, successful evaluation of food can be carried out and the product as such can be improved. Thank you.